Before we begin to look at the types of senses in particular, I want to introduce the ways that we classify sensory receptors. There are three ways. The first is by cellular type. The second is by location of that receptor in relation to the sensory stimuli that it interacts with. And the third is by the method of transduction. The first two are structural or anatomical ways of defining receptor cells and not the most prevalent way of classifying receptor cells. The third one, by transduction, is the functional or physiological classification scheme and really is primarily how we consider the receptor cells. When I ask you to consider what types of receptor cells we're talking about, I really mean by their transduction methodology. When we classify receptor cells by their cellular type, this really means to consider the microscopic anatomy. What type of cell is actually doing this? Truthfully, there's two types of cells. There are neurons and there are specialized receptor cells. However, we see three types of cellular classification here because the neurons can fall into two major groups, those with free nerve endings and those with encapsulated nerve endings. You've probably already looked at the free nerve ending, encapsulated nerve ending types of cells because that's something that would have been discussed within the integumentary system chapter of anatomy and physiology, or the skin chapter. Here we see representations of three major types of receptor cells. Part A of this figure has a neuron with free nerve endings. The dendrites are not encapsulated with anything. And part B shows a neuron with the dendrites encapsulated by another structure. There are several types of encapsulated nerve endings, which I'll show you in a little bit, but this is just a general representation of that. And then part C shows a specialized receptor cell. The example given here is a photoreceptor, a rod photoreceptor, which would be found in the retina. This figure demonstrates a couple of types of encapsulated nerve endings found in the skin. Notice in the bracket labeled dermis on the left side, there is a Meissner's corpuscle indicated. In the subcutaneous fat region down below, not next to the bracket, notice that there's also an indication, a label for a Pacinian corpuscle. Those are two types of encapsulated nerve endings that we associate with the skin. Those are the eponymous names, named after the person that discovered them. Often we refer to the Pacinian corpuscle especially as the lamellated corpuscle. Meissner's corpuscle goes by a few different names, sometimes mechanoreceptor type 2, those sorts of things, but there are various names for those two examples. The figure demonstrated a couple of slides before showing the different types of cellular receptors included the rod photoreceptor as an example of a specialized receptor cell type. This picture shows another type of receptor cell type, the taste cell. This is a rendition of a taste bud. You can see the opening at the surface of the tongue at the top, the taste pore. Saliva, including the chemicals to which these cells are sensitive, would enter through the taste pore and interact with components of the taste cells. The taste cells then release neurotransmitters directly onto the dendrites of a sensory nerve. So the black dot seen in the rather central taste cell, the one with the label on it in this picture, those black dots represent neurotransmitter vesicles. The neurotransmitters are released based on changes in the membrane potential of the taste cell, although it is not a neuron and is not generating anything like an action potential. The neurotransmitters released onto the receptor cell endings, or neuron endings there, will then initiate an action potential in that nerve. Now, since this is a taste bud, it's going to be interacting with the dendrites of either cranial nerve 7 or cranial nerve 9. Cranial nerve 7, the facial nerve, its sensory component is for taste of the anterior portion of the tongue. Cranial nerve 9, the glossopharyngeal nerve, is responsible for sensory reception of taste in the posterior portions of the tongue and areas of the soft palate. So while the receptor cell portion of this picture, gustatory afferent nerve, is not indicated here, it's either from nerve 7 or 9. The sensory ganglion that's indicated with the arrow there is going to be the ganglia associated with either of those nerves for the sense of taste. The second criteria that we can classify receptor cells on is that called location here. This is referring to the location of this receptor cell in relation to the stimuli that it's going to receive. There are three options here. The first is exteroreceptor, meaning any receptor in relation to external stimuli. And the second is interoreceptor, referring to any receptor in relation to interior or internal stimuli. That would seem to cover all possibilities, either inside or outside your body. However, proprioceptors or proprioreceptors are a special class that fall in between these two. Proprioception, the sense of your body's position and movement in space, is based on interoreceptor type stimuli, stretch of receptors or compression of joint capsules, Golgi tendon organs, stretch in the skin, those sorts of things. So they're internal stimuli of those organs, but in the sense that it's our body in relation to the external world. So proprioception doesn't easily classify as exteroreception or interoreception, and it has its own classification. Again, location, as with the cellular types of classification, are minor ways of classifying the cells. The more important one is the third, the functional transduction type. So this third type of classification for sensory receptors, transduction, is the functional classification and really the most important aspect of this. Now, the list that you see here is not necessarily exhaustive. We could certainly describe a few other types of receptor cells, but these are the major ones that you're likely to be introduced to. Now, the word transduction, or to transduce, really means at an etymological level to carry across. And so transduction is how a stimulus gets converted into the nervous system's electrical signaling coding, action potentials. So these are cells that take the stimulus and change them into the electrical activity of the nervous system. These could easily be neurons doing it themselves, such as the free nerve ending or encapsulated nerve ending cellular types, or they could be specialized cell types, such as photoreceptors or taste cells or something like that. The structural particulars of the cells is not important at this point. We're really just concerned about how the signal gets changed from one realm, the stimulus itself, into the nervous system signal, the action potential signal. Each of these types is based on a name constructed for the type of stimulus and the receptor cell that does that. For example, a mechanoreceptor is going to be a receptor that is sensitive to mechanical stimuli. I've already mentioned a couple of times stretch receptors, like in distension of the stomach or the bladder. Those are mechanoreceptors. Physically stretching out the tissue that includes that receptor is going to cause a change in the electrical properties of that cell. In the inner ear, there are some very important mechanoreceptors, which we'll talk about in more depth in relation to hearing and other aspects of the inner ear. And those are also mechanoreceptors, which might not seem quite so obvious, but really sound is a mechanical sense. You'll hear more about that later on. Thermoreceptors, it should be obvious, are going to be ones that are sensitive to temperature. There's basically two types of thermoreceptors, cold receptors and hot receptors. If the temperature that the receptor is being exposed to is below body temperature, then a cold receptor will respond to that. A hot receptor would not respond to a below body temperature response. Hot receptors are going to respond to things that are above body temperature. If you're interested, you could do a little extra investigation into thermoreceptors. There is an interesting effect that can be manipulated where you can fake out certain types of thermoreceptors to being responsive to the wrong type of thermal energy, so to speak. Photoreceptors, hopefully, are obviously light receptors. Chemoreceptors are chemical receptors. And there are a number of different types of chemical receptors. The other two entries on this list are really specialized chemoreceptors. But chemoreceptors involve a number of different senses, such as taste, the taste cells that I pointed out a little while ago. In the olfactory system, the olfactory neurons are chemoreceptors, et cetera, et cetera. Nociceptors are a special type of chemoreceptor. Noci is the root of the word, say, obnoxious or noxious, which means painful. And so nociceptors are pain receptors. Really, pain is a chemical sense. Anytime that there's tissue damage or the threat of tissue damage, there will be a release of chemicals that will stimulate nociceptors. If a tissue is physically damaged, if you cut your skin, for instance, the cells that make up your skin, when they're broken open, are going to release chemicals into the surrounding tissue. Some of those chemicals will be released because the cell's just been breached, and what's inside the cell will spill out. Also, tissues tend to respond to damage by releasing additional signals. We'll actually look at some of those signals when we get to talking about the immune system, because those signals that are released that are going to activate pain receptors are also going to be responsible for initiating some of our response to infection and injury. Osmoreceptors are a special kind of receptors also. They're chemoreceptors in the sense that we're talking about water as the chemical. So osmoreceptors are sensing the osmolarity or osmolality of the bodily fluids. One place where we see osmoreceptors is at the rear portion of the tongue and at the opening of the pharynx. They're receptive to how much water we're taking in, which is an important bit of information that we need to process in relation to digestive function. There are probably some other cells that could be classified in this list too. I've just given you these 6 as an indication of some examples, but this is not at all exhaustive. Consider this question. When you feel that you've reached an answer, hit the next button to go into the next slide, which will reveal the correct answer.